A very good morning to the students of class 12 science. In the last class, we have talked about vaccination and immunization. We have discussed about autoimmunity also. Clear? Autoimmunity means where our body immune system will work against our own cells. It will not be able to discriminate between the foreign cells and the body cells. So we have also mentioned about the different examples of diseases under autoimmunity. So in today's class, we will discuss about immunodeficiency. And under this topic, we'll learn about acquired immune deficiency syndrome, or we used to call it AIDS. Yeah, HIV/AIDS simply. So, what do you mean by immunodeficiency? What immunodeficiency means where the body immune system or the immune power of an individual slowly begins to decrease or starts to fall. Okay, so it's called means immunity, deficiency means decrease in the body immune power or immune system, or we can say in immunity. So under this one, you've heard about the HIV/AIDS, the human immunodeficiency virus that causes acquired immune deficiency syndrome, where the immune system of an individual will decrease. Clear? So first of all, HIV/AIDS, it's mainly a disorder of cell-mediated immune system. Now, what do you mean by cell-mediated immune system? If you remember or try to recall, we have learned about humoral mediated immune system and the cell mediated immune system. Humoral mediate was under the control of antibody formation, you can see that, and cell mediated immune system is totally dependent on the T lymphocytes or T cells. So therefore, AIDS is mainly a disorder of cell-mediated immune system of the body, where the HIV virus will directly go and infect the T lymphocytes or the T cells. Now, why do you think that whenever the HIV virus goes and attacks the T lymphocytes, why there is decrease in the body immune system? The name suggests immunodeficiency. There will be deficiency in the immune system of an individual. So whenever the HIV virus will infect an individual, then therefore that particular virus, once it enters the body, it will go and infect the T lymphocytes. Now you know that T lymphocytes or T cells is very important because it will stimulate the B lymphocytes for the formation of antibody, and that antibody will work against a particular pathogen, or we can say foreign materials or the foreign microorganisms. So over here, the number of CD4 cells or the number of helper T cells will decrease. So I mentioned that once this HIV virus will enter the body of an individual, it will go and affect the helper T cells or the helper T lymphocytes. So once the T cells will be infected, its number will keep on decreasing. Clear? The number of helper T cells will keep on decreasing, and as a result, the immune system of an individual will also decrease. Clear? Because it will be no longer available to help or to stimulate the B cells or the B lymphocytes for the formation of antibody. So there will be reduction in the number of helper T cells, and at the same time the body immune system of an individual will also decrease. And therefore when the immune system of an individual will decrease, and it will result in the loss of the natural defense against viral infection. That means an individual will not be able to fight against a common infection also, once the immune systems will slowly deteriorate or decrease. Clear? So discovery of HIV or AIDS, kindly go through the page number 467 from your textbook. One more time, page number 467. Go through the discovery of AIDS. Okay, so you can see over here it was first reported in 1981 in USA, and it was isolated and identified by, kindly remember this name, okay, Dr. Luc Montagnier in 1983 in France and by Dr. Robert Gallo in USA in 1984. Clear? The AIDS virus was named Human Immunodeficiency Virus HIV in 1986 by the International Committee on Taxonomy of Viruses. So kindly go through the discovery of AIDS from your textbook, page number 467. Clear? So now we'll go with the structure of HIV virus that causes AIDS. Okay. So from textbook, you have to go with the figure number 8.20. Clear? So let us go through the structure. It's very easy. Clear? So kindly go through the diagram. So first of all, you can see this rounded structure, right? So therefore HIV is a rounded retrovirus. So HIV belongs to a class of viruses called retroviruses. Now retroviruses has got RNA as genetic material. Remember, clear? Retroviruses has got RNA as genetic material. Kindly remember this thing. Okay, it has got RNA as genetic material. Clear? So first of all, the HIV is a round retrovirus. Clear? With two single strands of genomic RNA. Listen very carefully. With two single strands of genomic RNA. I'm saying that retroviruses has got RNA as genetic material, and RNA is single stranded. DNA is double stranded, whereas RNA is single stranded. So therefore HIV is a rounded retrovirus with two. So can you see over here? This one is the first you can consider, and this is the second one. So with two single stranded RNA. So this is the RNA, single stranded RNA. So these are the two single stranded RNA which are present in case of HIV virus. Then next thing, it has got the enzyme called reverse transcriptase. So you can see over here, small dot over here, this is the enzyme reverse transcriptase over here. Okay, this one, this is the reverse transcriptase. Small dot one is there, which is the reverse transcriptase. So this is the two genomic RNA. Small dot over here, this is the reverse transcriptase. Now what is the function of reverse transcriptase? The function of enzyme reverse transcriptase is during the process of reverse transcription. Now what is reverse transcription? It is the process of formation of DNA from RNA. Clear? Because this particular HIV virus has got RNA as genetic material in the thing. It has got RNA as genetic material. So from this RNA, there will be formation of DNA. And for the formation of DNA from RNA, an enzyme is needed. And the name of that enzyme is reverse transcriptase. And the process where there will be formation of DNA from RNA is known as reverse transcription. Clear? So it is clear for you. So first of all, HIV virus is a round retrovirus which has got two single-stranded genomic RNA and the enzyme reverse transcriptase. The function of enzyme reverse transcriptase is to form DNA from RNA. Clear? Next thing, it has got two protein code also. So you have got the enzyme reverse transcriptase, then you have got two protein codes. So can you see over here these round structures? These round structures are the protein code. Okay, this is the inner protein code and this is the outer protein code. The inner protein code, that means this one, okay, this particular structure, this inner protein code is called P24 or P24, and this outer protein code, this one, okay, this outer protein code is called P18, P18. Then we have got the outer envelope. Clear that the entire structure is covered by the envelope. So one more time, structures, we are just talking about the structures of HIV virus. So HIV virus belongs to a class of viruses called retroviruses, and retroviruses, they have got RNA as genetic material. So therefore HIV has also got two single stranded genomic RNA. It has got the enzyme reverse transcriptase, which will help in reverse transcription, that will help in the formation of DNA from RNA. Then we have got two protein code, outer protein code and the inner protein codes. The inner protein code is also called P24 and outer protein code P18. Then we have got the envelope. Clear? So envelope, that means this particular structures, okay, this particular structure is called envelope, this outermost circle. Clear? This is the innermost circle, in that envelope we can say, and this is the outer envelope. Clear? So therefore envelope is formed of lipid bilayer. Bilayer means two layers. So envelope is composed of two layer. Clear? And is mainly composed of lipids. So we have got the lipid bilayer. So this is the inner lipid bilayer and this is the outermost lipid layer. So we have got two layers. It's mainly composed of lipids. Clear? So then we have got the projectine nuclei structures, which is composed of glycoprotein spikes. That means these spikes we are talking about. Okay, we are talking about these spikes. So these spikes are composed of glycoprotein. Clear? And these spikes has got pericels, which is mainly composed of glycoprotein. Clear? So one more time, we will briefly discuss the structures of HIV virus so that it will be easier for you. Okay, first one, HIV virus is a round retrovirus having two single genomic RNA, these two single stranded genomic RNA, then the enzyme retroviruses, then we have got protein coat. Clear? In the structure is covered by an envelope. This envelope is composed of lipid bilayer. So these particular structures are the lipid bilayer. Then from this particular envelope, we will find certain projection or a projecting nuclei structure, which is called glycoprotein. No, what the glycoprotein spikes. So this is the general structures of HIV virus. We can say AIDS virus. Can you remember the word retroviruses? We just got RNA as genetic material. Clear? Okay. Next one, mode of transmission. So how are this HIV virus transmitted from one individual to another individuals? So this one is also there in the page number 467. So simple. First one you have got over here, sexual intercourse, because this HIV virus are present in the hymen, present in the semen, 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 and also in the vaginal fluids. Clear? Of an individual organisms. Suppose if fetus is infected, so having an unprotected sexual intercourse, or if one of the partner is infected and having an unsafe sexual intercourse or unprotected sexual intercourse will lead to the transmission of HIV virus from an infected individual to the healthy individuals. Next one, the HIV virus can also be transmitted during blood transfusion. That means there will be when the blood will be donated from the one individual to another individuals. That's called transfusion. Okay, so if the blood contains the HIV virus, the blood which is to be transfused, if that particular blood contains the HIV virus, then the recipient of that blood will also become infected with HIV virus. Clear? The next one is shared needles. If the needles or the syringes which we use in case of hospitals or any other syringes which are used by the dentist or any other medical practitioner, if those syringes or needles are not well sterilized, clear, and if those instruments are being contaminated by the HIV virus, and if they are used without proper sterilization, then in that case also the HIV virus can be transmitted. All right, mainly in case of intravenous drug administrations, we use those syringe or needles. So it has to be properly sterilized before using. The HIV virus can also be transmitted from the mother to the child. If the mother is HIV infected or HIV positive, in that case also the HIV virus will be transmitted from the mother to the child. Okay, during organ transplantation, now you know what is organ transplantation. If one of the organ is like is functioning not properly, dysfunctioning or malfunctioning, in that case we can go for the organ transplantation. In that case also, if an individual is HIV positive and if we are using the organ of that particular individual, then those who is receiving that particular organ will also become infected by HIV virus. So during organ transplantation also HIV virus can be transmitted. At the time of parturition, that means during the time of the childbirth, right, if the mother is infected, then the virus will be transmitted from the mother to the child. Clear? And during artificial insemination also, where we used to transfer the sperm, all right, those sperms which are being stored, and if the stored sperms are being injected into the reproductive tract of the female, and if that sperm contains the HIV virus also, so at that time also during artificial insemination also there can be transmission of HIV virus. So during sexual intercourse, during blood transfusion, using those set needles or syringes, during organ transplantation, during artificial insemination, and during parturition also, the HIV virus can be transmitted from an infected individual to a healthy individual. Clear? So this is the mode of transmission. So kindly go with the page number 467. The most important thing, mode of action of AIDS virus. Clear, because we are saying that once this HIV virus or the AIDS virus will enter the body of an individual, it will go and infect the T lymphocyte or the T cells, so that it will no longer be available to help the B lymphocytes for the formation of antibody, and as a result, the immune system of an individual will keep on decreasing. Okay, so now we will just see the mode of action of this AIDS virus, how they are replicated, how they are multiplied within the body of an infected individuals. Now we are going to discuss about that. Okay, so from your textbook, kindly go with the figure, go with the page number 468 and figure number 8.21. So here we are going to learn about the replication of HIV virus or the retrovirus. So before that, what is replication? Okay, we have to know the term replication. Replication means where there will be multiple productions, we can see in this case, okay, where there will be multiple productions of the daughter DNA from the parental DNA, that we learn in the chapter human genetics also. Clear? We will learn with DNA replications, where from a single parental DNA there will be formation of multiple copies of daughter DNA. Simple, that's called replication, to replicate, to produce the carbon copy. Clear? Just remember these things. In details we will learn in the chapter human genetics. All right, okay, now let's go with this topic. So how does this HIV virus or the retrovirus will multiply within the body of an infected individuals? So remember that retrovirus has got RNA as genetic material, and from this RNA they have to form the DNA by the help of enzyme reverse transcriptase. So please keep these points in your mind. All right, so first one, the virus enters into the macrophage after getting into the body of a person. So first thing is that this HIV virus or the retrovirus needs to enter into the body of a person. So first thing, the virus needs to be entered into the body of an individual organism. Clear? So once it will enter the body, then it will go and enter into the macrophage. Remember this thing. So first thing is that the virus has to infect the normal cell. Clear? This is the retrovirus having RNA, clear, and the viral protein code. So it will infect the normal cell. Once it will infect the normal cells, clear, once it will enter the body of an individual, it will go and enter into the macrophage. Clear? So now the viral RNA has entered into the macrophage of a particular person being infected. Hope you are getting it. So first thing, the virus needs to enter the body of a person. After entering, it will go and infect the macrophage. All right, it will go and resides in the macrophage. Now, within the macro, let's say inside the macrophage, the RNA of the virus replicate to form DNA. Okay, so you can see over here viral RNA. Viral RNA is introduced into the cell, so it is within the cell of a person, mainly in the macrophage. Now that viral RNA, okay, the viral RNA, the RNA of the virus will form the viral DNA. So how does the viral RNA is converted into viral DNA? For this particular process, for the formation of DNA from RNA, viral DNA from viral RNA, we require a protein, all right, sorry, an enzyme. The name of that enzyme is reverse transcriptase. Clear? So reverse transcriptase is the name of the enzyme that will help in the formation of viral DNA from viral RNA, and this particular process is called reverse transcription. Clear? So the viral RNA has entered the cell, has entered the macrophage. Once it enters the macrophage, it undergoes reverse transcription, where it will form viral DNA from viral RNA by the help of the enzyme reverse transcriptase. So now there's formation of viral DNA. Clear? Now, after the formation of viral DNA, this viral DNA, okay, this viral DNA will get incorporated. All right, this viral DNA gets incorporated into the host genome. So now that viral DNA will become the part of our genome. Clear? Remember. So viral DNA will become the part of our genome, and whenever our DNA multiplies, alright, whenever DNA replicates, at the same time, those viral DNA will also multiply or will replicate. Clear? Okay, getting it? First thing, virus will infect the person. It enters the body of an individual. After entering the body, it will go and reside in the macrophage. In the macrophage, those viral RNA will form viral DNA by the help of enzyme reverse transcriptase by the process known as reverse transcription. Once there will be formation of viral DNA, that viral DNA will become the part of our genome, so that whenever our DNA multiplies or replicates, at that time the viral DNA will also replicate. Okay, so the viral DNA incorporates into the host genome. Now that viral DNA will become the part of our genome. You can say by the host DNA, that means our own body DNA, and this is the viral DNA. So the host DNA, whenever it will replicate to produce the daughter DNA, at the same time this viral DNA will also replicate. So when the viral DNA will replicate, all right, then new viral, you can see over here, then new viral RNA is also being produced by the infected cells. Clear? So look over here. So viral DNA gets incorporated into the host cell DNA and direct the infected cell to produce the virus particle. So this viral DNA becomes the part of the host genome, and whenever this host DNA will replicate, the viral DNA will also replicate and will produce the more amount of viral RNA or the viral particles. Clear? So you can see over here that the macrophage is playing an important role in the production of more viral particles or viral RNA, because viral or HIV virus will enter the macrophage and it will keep on replicating. It will keep on producing multiple copy of viral RNA. Okay, so that's why the macrophage is also known as HIV factory. Why? Because it will continue to produce the virus. Clear? So because virus will enter the macrophage and macrophage will help in the rapid replication of this viral DNA or the viral particle to form the viral RNA or new viruses. Now, since macrophages are continuously producing the virus particle, it acts like a HIV factory. And at the same time, those viral RNA which are produced, okay, those viral particles which are produced at the same time, they will go and infect the T cells, okay, simultaneously. Once they are produced simultaneously, they will go and infect the helper T cells or the T lymphocytes. Clear? And will produce the more progeny. So new viruses are being produced, and once these viruses are being produced, they will go and infect the helper T cells or the helper T lymphocytes, and at the same time, more viral DNA are being produced. Now, once more viral DNA are being produced, they will go and infect the T lymphocytes. Okay, because these progeny viruses, they are released into the bloodstreams and they will go and attack the other T lymphocyte. That means the number of T lymphocytes keeps on decreasing. Why? Because they are continuously infected by the new viral particles which are being produced. Okay, so the new viral particles which are being produced, they are released into the bloodstream, then they will go and infect the new T lymphocytes or the T cells. As a result, the number of T cells will keep on decreasing. Alright, so when the T lymphocytes are being infected by the viral particles or by the viruses, there will be decrease in the number of T cells or the T lymphocytes. Then T lymphocytes will not be available to help the B lymphocytes for the formation of antibody, and when there is no formation of antibody, the immune systems usually decreases. All right, it will usually, the power or the immune system usually becomes very weak. It will not be able to produce the antibody that attacks the invading pathogens. Clear? So it's very easy. So first of all, remember that retroviruses or HIV viruses, they will enter the body of an individual organism. Once it enters the body of an organism, it will go and enter inside the macrophage. Within the macrophage, the viral RNA will form the viral DNA by the help of enzyme reverse transcriptase. Now that viral DNA will become the part of the host genome. So whenever the host genome will multiply or replicate, the viral DNA will also replicate and produces more viral particles. Now since macrophage will help in the production of more viral particles or more viruses, it acts like a HIV factory. Clear? And simultaneously at the same time, those newly produced viruses will go and infect the helper T cells or the T lymphocytes. So more number of viruses will be produced, they will enter the bloodstream, they will go and infect the T lymphocytes. And when the number of T lymphocytes will keep on decreasing due to the infection by HIV viruses, then therefore it will no longer be available to help the B lymphocytes for the formation of antibodies. So our body will not be able to produce the antibody, so that our immune system will become very weak and will not be able to fight against the invading pathogens. And after that, what happens is that an individuals with weak immune system, with weak immune power, will no longer be able to fight with the minor infection also, that can lead to the death of an individual organism who are infected by the HIV virus. So kindly go through the mode of action of AIDS virus. Go from textbook, page number 468, figure number 8.21. Clear? Okay, next thing, symptoms of HIV AIDS. Yeah, I've got simple fever, okay, cough, slight but recurring diarrhea that will keep on reoccurring again and again, rashes on the skin, and there will be weight loss. Okay, in rare conditions there can be lung infections. You can develop cancer that affect the skin and the lining of the blood vessels and spread throughout the body. Okay, those who are infected by HIV virus, they are susceptible to unusual bacterial and fungal infections. Why? Because the body immune systems will be very weak, so they cannot fight with even a simple infection also. There can be heart disease, diarrhea, ulcerating infection of the mouth or of the genital opening like anus. Okay, AIDS has a long incubation period. Now, what is incubation period? Incubation period means the time interval or the time period between the entry of viruses into the body and the appearance of the first symptom of the disease. And this particular incubation periods in case of HIV infection, it ranges from few months to several years, mainly from 5 years to 10 years. It can be the incubation period of HIV viruses. It can be within from a month to a year, 5 years to 10 years. But once the symptoms of the disease starts occurring, and usually within a year we can say the patients cannot survive. There will be death of an infected individuals. Though there are certain drugs which are available as a cure for the HIV/AIDS, but those particular drugs are simply given to increase the lifespan of an individual organism. Right? They are not the permanent cure for the diseases. So that we are going to see the name of the drugs. Now, how to detect the HIV/AIDS patient or how to diagnose screening. So those who are infected by these HIV viruses or by the retroviruses, their body contain antibody. Okay, so we have to detect the presence of these antibodies against the HIV viruses, those who are infected by HIV. Clear? So this can be done by taking the blood serum of an individuals. Clear? And by taking the blood serums, we will try to find out whether this antibody is present in the serum or not. So we will try to find out the HIV infection in a particular individuals. The most important thing over here is the test. Okay, main diagnosis of AIDS is mainly done by a test called ELISA test. ELISA, ELISA test. Can you remember the word? ELISA means enzyme linked immunosorbent assay. So ELISA test is mainly performed to diagnose the individual organisms who are infected or not by the HIV viruses. Then a positive ELISA, if the test results of ELISA comes positive, then for confirmation, okay, whether an individual is HIV positive or not, mainly for confirmation, another test is performed which is called Western blotting test is performed. We have the simple technique for the screening of AIDS patient. Kindly remember this thing, and over here is very important. Okay, ELISA is very very important. ELISA means enzyme linked immunosorbent assay. Treatment, a drug is there called azithromycin, and I have told you that this is not the permanent cure for the HIV infection. It's simply the drug that has increased the lifespan of AIDS victim or of an HIV positive individuals. And now there are different types of drugs which are under trial. All right. And mainly there is this professor Luc Montagnier of France. All right. He mainly told that those new drugs which are being discovered, all right, like aerosol, pentamidine, AP, which are under trial, if these drugs are given with combination with AGT, if the drugs are given in combination with AGT means azithromycin, then it will be very very much effective. Clear? So these particular drugs, azithromycin, this aerosol, pentamidine, are simply given to increase the lifespan of an HIV infected individual. They are not the permanent cure for the disease. Clear? So kindly go through the screening of HIV infection or HIV/AIDS, then with the treatment. Now we'll go with the prevention, how to prevent and how to control the disease. First one, there should be mass education among the people, so people should be educated about HIV/AIDS, right? Number two, only disposable needles and syringes should be used for injections. You know what do you mean disposables? Once they are used, it cannot be used again and again. So those type of needle series needs to be used. Insist the use of thoroughly sterilized equipment by the dentist or the doctor. That means any equipments which are being used by the medical practitioner, they have to be thoroughly sterilized. Next one, there must be routine screening of the blood donor. That means who are donating the blood, there must be routine screening of those individuals. Alright, so there will be routine screening of the blood donors, organ donors, the donor of semen for artificial insemination and growth hormone. Clear? And of females in high risk group who are pregnant or contemplating pregnancy. So those individuals need to be thoroughly screened or really screened for HIV infections. Sexual habits needs to be changed immediately. Need to stick to single sex partner or instead of having multiple sex partners. Before receiving blood, the blood must be screened for HIV infection, whether the blood is being infected by HIV virus or not. So it has to be there. Very important. Okay, so during blood transfusion, the blood needs to be screened whether that blood contains the HIV virus or not. It's very important. Last one, do not share a razor, toothbrush, and any other article that can be contaminated with blood, because if those articles are being contaminated by the blood, and if that individual is infected by HIV virus, then those who are using it, they also become infected by HIV virus. So go through the prevention and control of HIV/AIDS also. So today we have learned about the HIV/AIDS. We have learned about the name of the viruses, the retroviruses. We have learned about the structure of retrovirus or the HIV virus. Then we have seen the mode of action of HIV viruses, right? We have learned about the screening of AIDS. We have learned about the treatment of AIDS, though there is no permanent cure for the disease, but there are certain drugs which will increase the lifespan of the infected individuals. And we have talked about the prevent and control measures of HIV/AIDS. So this much for today. In next class, we will begin with the new topic. Thank you.